Hello everyone, so today we're going to do a semester 1 biology review, so hopefully this will be a nice quick truncated version of all or most of the important things that you went over for the Intro to Biology unit, Cells unit, Cell Function unit, and the DNA unit. So starting off with Unit 1, we talked about Intro to Biology, and this is just kind of an overcap of what biology really is, and biology is the study of life, anything that's living. All life between the simplest cell, or prokaryote, to life and its interactions with other living things and their environment across the biosphere. So we've kind of talked about hierarchy, atom makes a molecule, molecule makes a macromolecule, several macromolecules working together make the smallest unit of life, which is the cell. Those cells can make tissues, tissues make organs, organs make organ systems make organisms, organisms make populations, populations living amongst other species of populations make a community, several communities living near each other is an ecosystem, several ecosystems and combinations are biospheres, etc., etc. So biology pretty much studies the smallest little living thing under a microscope to, looking at it from a telescope in space, like zooming into areas on our planet. So topics within biology are zoology, which is the study of animals, botany, the study of plants, marine biology, which is the study of marine mammals, or animals, marine animals, and paleontology, which is the study of past living things or fossils. So anything that has ever been alive, there's typically a specific type of biology you could study to get a more zoomed in approach or content area for those topics. Then we have different fields of science, so not biology. There are chemistry, physics, geology, weather. There's all sorts of different kinds of soil sciences, all sorts of different kinds of sciences that don't exactly look at living things, but perhaps how those living things work through chemistry or the physics, etc. So if biology is the study of life, then we need to understand what the characteristic of living things are. If we don't know if something's alive or not, we don't know if it fits into the category of biology. So in order to consider being living, an object must meet certain criteria. And this criteria list is typically 5 to 7 criteria long. If you were in my or Miss Foy's class in the fall, we went over 7 different criteria, but minimally across the board, scientists are all able to agree on at least just 5. And the 5 that you're going to be tested on or that was taught specifically in this course are the ability to reproduce, respond to the environment, be made of cells, be able to grow and develop, and obtain and use energy. So those are the 5 things that we're going to talk about on the next few slides in more detail. All right, number 1, the ability to reproduce. So this is generally just spreading on genetics to future generations to increase genetic variability to help increase survival for your species. Okay, so if an individual is not able to reproduce, that doesn't mean that they're not living. So if I or someone I know is infertile and is incapable of having their own offspring, that doesn't mean that they're not alive. This reproduction criteria is just for the whole species as a whole. So even though if I may not be able to reproduce, my sister can, or my neighbor can, or someone on a whole different continent can, as a whole, humans as a species can reproduce and continue on to future generations to maintain them as a species. So this doesn't need to be an individual thing. It's just mostly a community kind of thing. Then we have the ability to respond to the environment. This doesn't mean that you need to be able to move or run away. We know that trees, when a man with an axe is running at the tree can't just pick up its roots and run away from, the danger, right? But just to respond. So flowers opening up when the sun is out, or trees losing leaves or changing color when it starts getting cold and the amount of sun during the day is reduced. Or alternatively, when spring comes around, the temperature increases, the amount of sunlight increases, and that causes the plants to respond by creating leaves so it could start photosynthesis. But of course, responding to the environment could literally be a rabbit running away from a coyote, or, a bird recognizing its hailing and getting out of, the hail and the damage that might cause it. So the ability to respond to the environment is one of the criteria to be considered living. Then we have be made of cells. And so cells are the smallest building block that is considered alive. Again, atoms, molecules, macromolecules, cells. Smallest thing that we consider is alive. A macromolecule doesn't fit the 5 criteria that we're talking about. Neither does a molecule or an atom. All right, but cells can respond to their environment. They can reproduce. They are themselves made of cells. They do grow and develop. They meet all of the criteria to be considered alive, so they're the smallest unit or building block. Then we have grow and develop. So multicellular organisms going from one cell to millions and creating a functioning body or organism. Okay, so you start off as a fertilized egg. So that is your mother's egg mixing with your father's sperm to make the very first cell that is you. That cell has all of the DNA you need to make you what you are and how you are. And then that cell is going to start dividing to create, more tissues and organs to make a viable baby. Okay? Unicellular organisms still also grow and develop, even though they remain one cell their entire life. They still grow larger and change functions in order to survive, and then in order for them to reproduce, they just make a copy of themselves, and then that makes a whole nother cell. Lastly, we have obtain and use energy. So this is to produce their own sugar into energy. So producers, plants, take energy from the sun to make their own sugar. Okay, that sugar is then sent to mitochondria to make energy for the plant to live. Consumers need to consume other organisms for their sugar and for their energy, and then to convert to energy. So a grasshopper is going to eat the plant. The grasshopper cannot make its own sugar, it needs to eat the plant for the sugar, which then its own mitochondria converts that sugar into usable energy. So in this image, the bird might eat the grasshopper. Again, the bird can't make its own sugar, it needs to eat the grasshoppers for sugar, which then it uses its mitochondria to convert that sugar into ATP. So it kind of goes throughout the chain like that. Okay, and how we obtain and use energy deals with our metabolism. If we have a much higher metabolism, we have more of a caloric need. So that bird might need to eat 30 grasshoppers a day or something to live because of its metabolism and its metabolic needs. So now that we've reviewed the 5 things that make something alive, we're going to talk about the scientific method. And the scientific method can be taught in a variety of ways. Again, sometimes there's just 3 steps, sometimes there's 5 steps, sometimes there's 7 steps, sometimes there's not a linear method. That's okay. But in this unit and how it was specifically taught to you through Buzz, they had 5 steps. And the first step would be to collect information and make observations. So if you want to, do an experiment where you want to grow tomatoes and you want to see, well, does adding fertilizer make my tomatoes bigger or make my plant produce more tomatoes? So I might collect information by looking things up on the internet, seeing if there's information there, and just generally making observations. Over the years, when I added fertilizer, did it give me bigger tomatoes or did it give me more tomatoes? Then you need to form a hypothesis. So this is a question with a prediction. Typically an if and then statement. If I give plants fertilizer, more tomatoes will grow. Okay? If and then. If I give my tomatoes fertilizer, more tomatoes will grow. Then I'm going to form an experiment. With this experiment, I need both plants conditions or both group of plants conditions. Ideally, you have multiples. Just doing it on 2 plants isn't really going to give you an accurate depiction. Okay, I want to have 20 plants that receive fertilizer, and I want to have 20 plants that do not receive fertilizer. And an important part with an experiment is to make sure that you have a control group and an experimental group. And across both of those, you're going to have the same things happen to both of the groups except for the experiment. So in my control group, which is under normal conditions, okay, I'm going to have my 20 plants that do not receive fertilizer. I'm going to give them a set amount of water, a set amount of sunlight, and a specific type of soil. My experimental group is going to have the same amount of water, the same amount of sunlight, and the same specific type of soil as the control group. The only difference between these two groups is my experimental group is going to have fertilizer added. That way, if there's any differences in the end of my study, I could determine that it was just because of that thing that I changed, just because of the fertilizer, not because one group got more water or more sun, because both groups got the same stuff. Then, I would like to retest and come to a conclusion. So if I were only able to do one plant each, I would retest until I had enough to have it be statistically significant, or I could just have a giant group at once, okay? So once I have my retested results or my giant group done at once, I will be able to come to a conclusion, and that conclusion is, well, yeah, if I do give my plants fertilizer, they did grow more tomatoes, okay? Then that's typically where an individual experiment ends. However, it is some people's goal to identify a law or a theory. Okay, and you can't really make a law or a theory off of just your experiment. You need to have lots of other people test their own experiments or similar experiments and come to the very same conclusion. Once you have pretty much across the board a bunch of different scientists in consensus or in agreement over if you do this, then this happens, then it may become a law or a theory. I accidentally wrote a law of theory. Sorry, it's meant to be an or theory. Let me write that. Can I do that? Nope. Okay, well, it kept going anyways. All right, so then we have the cell theory. In the cell theory, there's three main rules. Okay, in order for something to be even called a theory, remember, across the board consensus and research needs to find that these things are true. So for the cell theory, all things must be made of cells. Cells are the basic units of structure and function in living things. And all cells come from pre-existing cells. Okay, so as far as all living things are made of cells, well, we know that the cell is the smallest thing that is considered to be living. It meets all those 5 criteria. Cells are the basic units of structure and function. Cells work together to make tissues. Tissues work together to make organs. Okay, so without cells, we wouldn't have tissues or organs or organisms. Okay, and then all cells come from pre-existing cells. Cells don't just spontaneously appear. Cells go through a process where they copy their DNA. They go through mitosis and split, and then you have 2 identical cells, of which one of those cells might differentiate or just continue being another skin cell if the parent was a skin cell, things like that. Makes things like lipids, steroids, hormones, things like that. Okay, so the smooth ER will make lipids and steroids, hormones to be shipped outside of the cell. So this could be cell signals for other cells to realize something. Hormones, that way we make sure we have, the right kinds of estrogen or testosterone or adrenaline, things like that. Then we have the lysosome, and the lysosome is basically a big garbage can, okay? It basically breaks down excess or worn out cells or worn out parts, okay? So if it realizes, oh, this mitochondria is towards the end of its leg, it's being replaced by another one, we're going to break down this mitochondria and we're going to send it outside the cell in a nice little garbage can package. The lysosomes break down and bring the junk outside of the cell. Lysosomes are also really important as far as like detecting and breaking down viruses within the cells. Once they're able to capture it, they kind of break down the viruses and spit them out as garbage. The last thing that we're going to talk about that are in both plant and animal cells are nucleuses. Nucleuses store the DNA, and so the DNA always lives in the nucleus. And so the nucleus is basically the user manual or the instruction manual for the rest of the cell, because the DNA lives in there. Organelles that are specifically found in plant cells, not in animal cells, are cell walls, which go on the outside of the cell membrane. So both the plant and the animal cell, in the image to your left, has a cell membrane, okay? But only the plant cell has a cell wall on the outside of that membrane. And that cell wall provides structure for the plant. It's what allows trees to be hundreds of feet tall, but, animals to only be a few, 16 feet tall or whatever a giraffe is. Then plants also have chloroplasts. Chloroplasts are filled with chlorophyll, which is what gives plants their green color. And the chloroplast is what allows plants to take in sunlight and make their own sugar. Okay, so both plants and animals have mitochondria to convert sugar into energy, but only plants have chloroplasts. So animal versus plant cells. Again, the last image showed an animal cell versus a plant cell. Just wanted to, hone in on plant cells have cell walls and chloroplasts versus animals and plants share all other organelles. So now we need to know the difference between eukaryotic versus prokaryotic cells. Eukaryote, euc, technically stands for true, and yote stands for nucleus. So eukaryotes are true nucleuses, meaning they have a nucleus. They also have other membrane organelles, so mitochondria, endoplasmic reticulum, vacuoles, Golgi apparatuses. Those are found in eukaryotes. However, prokaryotes are a little bit more simpler, okay? They mean before nucleus. Pro means before, karyote means nucleus, okay? And they lack membrane-bound organelles, so they basically just have DNA floating around and then some ribosomes. Ribosomes aren't membrane-bound, but those are the only things that are found within prokaryotes, just loose DNA called a nucleoid and ribosomes. And often prokaryotes have things like flagellums and cilia to help them move around, because typically eukaryotes are multicellular, so they have enough cells to have things like bones and muscles to let them move around. But prokaryotes are typically, single-celled little guys, and in order for them to move around and respond to their environment, they use their flagellum or cilia to move. Alright, so now we're going to talk about homeostasis. So homeostasis is basically keeping internal conditions stable. So this could be something like body temperature. Your body is able to maintain homeostasis by keeping your body temperature right around 98.6. Some people are happy at 97, some people are happy at 99, but there still reaches a point where your body does not function correctly. Your organs can start shutting down if you get too hot. 101 is a fever for pretty much anyone. 95 or 94 degrees is very cold for some people too. So if your body was not able to maintain homeostasis, you could get too hot for your organs to function, or too cold and you might get hypothermic, which is too cold for your organs to function. So your body needs to, work in conjunction with this metabolism and other nutrients to make sure that your body stays within good conditions. This works the same way with, blood sugar. For the most part, most people's bodies are able to create enough insulin to make sure your blood sugar is at a good level. Unfortunately, there are individuals that have things like diabetes where they are unable to do that, and therefore they need to monitor their sugar or take additional medication to make sure that their blood sugar levels are good. Homeostasis can also be, hormones, so the amount of estrogen or oxytocin or testosterone in the body. Having the right amount keeps you happy and healthy. Having the wrong amount or an unstable amount can cause, your body to react poorly or differently or make you cranky, things like that. Okay? This can also work for physical things. If you get a splinter, your body will try to maintain homeostasis by keeping that foreign invader, the splinter, out. So it's going to do what it can to kind of push that splinter out. So as long as you're not stepping on it constantly, in theory, your body will push it out on its own. Within this unit, we also kind of talked about permeability, specifically in regards to the cell membrane. So the cell membrane is a lipid bilayer. Okay, and it allows certain things through and doesn't allow certain things through. So the three terms that I want to talk about here is permeable versus semi-permeable, that I left off this list, sorry, and then impermeable. So permeable means to allow through. So permeable is literally like an open door. Anything can walk in, anything can walk out, as long as it's not the size of an elephant. Okay, then we have semi-permeable. And let's think of like a window screen, or the screen on a window. It's got little holes in it, okay? So it allows oxygen to come in and out. It allows very, very small bugs, maybe little itty-bitty gnats or ants to come through, but it doesn't allow things like bees or bigger flies or birds to come through, okay? So it's semi-permeable. Some things are allowed in, some things are kept out. Then we have impermeable, which means unpenetratable. Okay, so it's basically a big solid wall. Nothing can come in and nothing can come out. So again, these are talking about things for the cell membrane. So the next part of this, we're going to talk about how things go through the cell membrane. Okay, we have diffusion, which is things going from high concentration to low through the cell membrane. It uses no energy to do this. And the things that go through with diffusion are small, nonpolar, and not charged. So they're literally able to just wiggle right through the semi-permeable cell membrane. However, if some things are large, charged, or polar, then it will need facilitated diffusion. And facilitated diffusion is again going from high concentration to low without the use of any energy. But these large, charged, or polar objects need to go through a tunnel to get into the cell to get into the cell. And that tunnel is a protein. Okay, so facilitated diffusion is large, charged, or polar things using a protein channel to get inside the cell. Okay, differentiating diffusion versus facilitated diffusion. Diffusion is just small things squeezing right through the cell membrane, going from high to low concentration. And with facilitated diffusion, it's large things going through a protein channel, going from high to low concentration. Then we have osmosis. Not the movie Osmosis Jones, but I guess you could kind of learn what osmosis is from the movie. But anyways, osmosis is just water going from high to low concentrations. So it doesn't quite fit underneath the diffusion or facilitated diffusion category because osmosis is only in regards to water passing through, not the millions of tiny little itty-bitty particles or larger particles that would go through with diffusion and facilitated diffusion. Then we have active transport. And active, in order for you to be active, you need to use energy, right? If you were to just kind of lay on the couch, you're not using much energy. But if you were to want to go be active and bike around your neighborhood, you're going to have to use energy. So active transport does use energy. And because it's using energy, it's going from low concentration to high. So this is opposite than the other ones. You're trying to shove more crap into your suitcase versus just letting it spill out all over the floor. And in order for you to shove more crap into your suitcase, you need to use some energy and some gumption and lots of hands and maybe a sibling to sit on the top of the suitcase so you could zipper it closed, okay? So active transport uses energy to make molecules go from low to high. So the next part of the unit, we talked about dehydration synthesis and hydrolysis. You guys did very well on this in the bell work, so hopefully I won't chat your ears off too much. But dehydration, D means without, hydration means water. And then synthesis on its own means to put together, to make. So the definition of dehydration synthesis is to put together by removing water. So you're dehydrating it, you're taking water away, but because you're taking water away, it allows the synthesis of something to come together. Then we have hydrolysis. Hydro means water, lysis means to break. Okay, so hydrolysis means to break with the addition of water. So by adding water to something, it causes the bonds to get disrupted and they could break apart. So in this part, we started talking about macromolecules. We have carbohydrates, we have lipids, we have proteins, we have nucleic acids. Okay, but carbohydrates and proteins and lipids all go through their own dehydration synthesis and hydrolysis. Okay, so what are carbohydrates? So they're sugars from breads, noodles, potatoes, rice, natural sugars found in fruits or added sugars, things like that. And what do carbohydrates do? They're short-term energy. Again, if you were to go and eat a crap ton of sugar or 3 cans of energy drink, you would have a lot of energy that you would immediately feel like you need to burn. Okay, so the function of carbohydrates is short-term energy, and sometimes it can be stored for about 24 hours, and that's stored in the liver in the form of glycogen. Okay, so you could either use this energy right away or kind of use it within the next day. Otherwise, it gets stored as fat, and fat is long-term energy. So there are 3 different kinds of carbohydrates. There's monosaccharide, mono meaning one, saccharide meaning sugar. So that's a single sugar. So something like glucose. Okay, it's alone. Then there's disaccharide, di meaning 2, saccharide meaning sugar. So that's 2 individual sugars combined to make a disaccharide. Then we have polysaccharide, poly meaning more than one, and saccharide meaning sugar. So the bottom right image is a polysaccharide called glycogen. It's several glucoses glued together or bonded together. Okay, so dehydration synthesis is where you remove water, and that causes these glucoses to stick to each other to create glycogen. Or you have hydrolysis, where you add water, and that causes these polysaccharides to break apart into disaccharides or monosaccharides. Now we have lipids. Again, another type of macromolecule. So what are they? They're typically fats or oils. Okay, their function of lipids is long-term energy storage. Again, they're fats, so fat is hard to get rid of on the body, and so that's a long-term energy storage. You have to be in a calorie deficit in order for your body to start breaking down fat, converting it into sugar to then use as energy. But other functions of lipids are insulation and steroids in the form of hormones and cholesterol. Not like the kinds of steroids that particularly make you super buff and angry and potentially have testicular shrinkage. Okay, steroids can literally be like specific hormones that your body needs, or cholesterol, be it good or bad cholesterol. So again, the main functions of lipids are long-term energy storage, insulation from the cold, and steroids. So the main kind of lipid that we talk about is triglycerides. There's 3 triglycerides, which basically means glycerol. Okay, so triglycerides is a singular glycerol, sugar, and 3 fatty acids in combination. So of the different kinds of fats, you've probably heard of things called saturated fats and unsaturated fats. I spelled fat wrong for unsaturated. Anyways, saturated fats are solid at room temperature, so things like butter. Therefore, they kind of clog arteries and are unhealthy. So if it's solid at room temperature, that means that it might be a little bit solid in your veins, okay? And that can cause clogs. Then we have unsaturated fats. Unsaturated fats are the ones with the kink in it because of the double bond. And these are liquid at room temperature, things like oils, okay? So you can have olive oil or vegetable oil, that's liquid at room temperature. This is considered a healthier fat because it tends to create less clogs in arteries and things like that. But of course, you need everything in moderation. So lo and behold, with lipids, excess calories get stored as fat. If you eat way more food than you burn throughout the day, or way more calories than you burn throughout the day, that's going to get stored as fat to be used later. When you burn more calories than you consume, the triglycerides get broken down through hydrolysis to be made into usable energy. So if you eat 12,000 calories a day, but you walk 3 miles a day and exercise and burn technically like 1,500 calories a day, your body's going to reach for that fat for energy to fuel your body, and that's how you start losing fat and losing weight. Then we have proteins, which is a type of macromolecule. So what are proteins? Proteins are basically amino acids that are found in meats, eggs, beans that act as the building blocks to make a protein. Okay, so when you eat something, you eat meat, it's the protein, but our body breaks it down into amino acids. So the functions of proteins are to transport things in and out of cells. So for transport, the functions of proteins are to make other proteins, muscle contraction and formation, things like that. So amino acids have all the same components except for the R group. So if you look to the image to the right, that is the chemical formula for an amino acid. And literally all of those amino acids, I think there's about 20 or so of them that are used within DNA to make protein, have the same components. They've got their nitrogen with 2 hydrogens attached. They've got their carbon with an oxygen and an OH attached. They've got just a simple hydrogen hanging out there. But the main difference here is this R group. Okay, so the R group is kind of like the side piece. It gets changed in and out depending on what the person feels like for the day. But the R group, depending on what is attached there, is what leads to the specific amino acid. So at dehydration synthesis in proteins, you have multiple amino acids being connected to each other. When you have multiple amino acids being connected to each other, it creates a polypeptide chain. And that polypeptide chain will, once it's done being created, will eventually start folding and twisting and contorting amongst itself to make a very specific protein that will have a very specific function. Then we have hydrolysis, the breaking apart of amino acid chains. So when organisms eat something with protein in it, it's a polypeptide, and it needs to be broken down into individual amino acids in order to be used to make more proteins, to help fuel the body, things like that. Also, sometimes proteins just age. They don't function correctly or are very old. So the protein can get broken down, and its parts can be recycled to make a new protein. Yay, recycling. All right, so now we have photosynthesis. So photosynthesis is the plant taking in light energy and making it into sugar. Not into energy, but into sugar. Photosynthesis takes place in a chloroplast. Again, this is one of those specific organelles that are only found in plants. And so below here is the unbalanced equation and the balanced equation. So the unbalanced equation is CO2, which is carbon dioxide, plus H2O, water, plus sunlight and energy, to then yield or make C6H12O6, which is a sugar, and oxygen as a byproduct that we very happily use. In order for this equation to be balanced, if you really like balancing equations because it's important for chemistry, this is think of it as kind of like the devil's number. There's a 666, right? And there's going to be a 6 in front of everything except for sunlight and the sugar. Okay, so we're going to have 6 carbon dioxides plus 6 waters plus whatever sunlight and energy the plant's getting, and that's going to yield a single sugar and 6 oxygens. Okay, so that's the balanced equation. With photosynthesis, there are some important scientists, one of which is Han van Helmont. Okay, that's a Dutch physician who discovered photosynthesis by planting a willow tree and then weighing the soil after it grew a bit to discover that the plant must have gotten energy from the sun versus the soil. If the plant was getting energy and sugar because of the soil, you would assume that the soil would weigh less, but it weighed exactly the same as before he grew the plant, and therefore he determined that the plant was able to grow because of the sun versus the soil. Then there's a man named Joseph Priestley, and he discovered that plants change the composition of air. Okay, so taking in carbon dioxide and putting out oxygen. Kind of cool. All right, so in order for photosynthesis to happen, plant cells need to have the organelle called a chloroplast. Okay, so the chloroplast image on left is a little green organelle. It's got an outer membrane, an inner membrane. Inside the chloroplast itself, it has stacks of thylakoids. This is where the chlorophyll, the green pigment, is found that makes plants green. This is where the light reactions take place. And then we have the stroma, which is the empty space or the fluid within the chloroplasts where the dark reactions take place. So again, in order for photosynthesis to occur, we need chloroplasts, and they have a two-part reaction in order to create their sugar and their oxygen. The first reaction is a light reaction. It takes in energy from the sun, as far as sunlight, as well as water to produce the byproduct of oxygen, and it also creates some energy to use in the next process, makes some ATP and NADPH. But these charged batteries do not leave the chloroplast. These charged batteries are used to energize the next process, which is the dark reaction. So the dark reaction is where carbon dioxide comes in, and then it uses our ATP and NADPH to create glucose in the form of C6H12O6. The ATP and the NADPH, once they are used as their batteries, turn into their uncharged battery form, which is ADP and NADP. Okay, so these are our uncharged batteries, and they just kind of float around within the chloroplast to then be recharged again in a later light reaction. So a lot of things are being recycled. So once we have sugar, plants go through cellular respiration. Or once animals or consumers eat sugar, they also go through cellular respiration. And cellular respiration is taking energy from glucose or sugar and breaking it down to get ATP, adenosine triphosphate, which is a chemical energy for the cell. And this takes place in the mitochondria. Both plants and animals have mitochondria, but only plants have chloroplasts. And so cellular respiration is literally the opposite equation of photosynthesis. Okay, so instead of taking in light or carbon dioxide, instead of taking in H2O and taking in energy from the sun, those are actually pretty much the products in this. So in cellular respiration, we start off with C6H12O6 because we've either eaten sugar from another thing, or if we were plants, we made our own sugar, and then we take in oxygen from the atmosphere. With that, we go through a chemical reaction that then spits out carbon dioxide, spits out water, and then spits out ATP to be used by the cells and the rest of our organs. Okay, so you know as a human, when you breathe out CO2. If you were also to breathe out on a clear glass, you would notice that condensation forms on the glass. That's because we're breathing out water with the carbon dioxide. Okay, so those are our byproducts, things that leave our body, leave our cells. And then we get to keep the ATP, or the energy, in order to run our bodily functions. So there's 3 main steps in total to yield 36 ATP. First step is glycolysis. This actually happens right outside the mitochondria. Sugar's a little bit too big to fit inside the mitochondria, so the cell needs to go through glycolysis to break the glucose up into 2 smaller pyruvates that do fit into the mitochondria. Okay, This overall creates 4 ATP, but only 2 ATP gets released for usable energy. In order to even go through glycolysis, you need to use 2 ATP. So when it creates 4, it actually keeps 2 ATP to do glycolysis again later, and then it spits out 2 ATP to be used for usable energy. So once the pyruvates are now small enough to fit into the mitochondria, it goes through the Krebs cycle. And this uses the 2 pyruvates to make 8 NADHs, 2 FADH2, and just 2 little measly ATPs. This is also where we get our byproduct of CO2. Okay, so the NADH and the FADH2 will be used in the electron transport chain, and then the 2 ATPs get sent out of the cell for use. Okay, so the Krebs cycle takes in the pyruvates, makes a bunch of energy, but only 2 ATP gets sent out to the cell for use. Then we have the electron transport chain. This is where the majority of the ATP is made. So it uses all that NADH and FADH2 that was made in the Krebs cycle, and then it produces 32 ATP. So, glycolysis just made a net of 2 ATP, Krebs cycle made 2 measly ATP, and then the electron transport chain makes 32 ATP. But we wouldn't be able to make 32 ATP without the first 2 processes. Then there's something called anaerobic respiration. So this means without oxygen. Only 2 ATP are made in anaerobic respiration versus 32 or 36, but that's not ideal, and it only happens when the body is really stressed from too much exercise and not getting enough oxygen. So ideally, you want to make a sugar into 32 ATP. You don't really want to make a sugar into just 2 ATP. Almost done, guys. Okay, now we've got mitochondria. We'll dig more into this little organelle. Again, it's found in all eukaryotes. It's found in both plants and animals. The mitochondria takes the sugar and converts it into usable chemical energy called ATP. And the 3 steps is glycolysis happens outside the mitochondria. Then the Krebs cycle happens within the matrix. So let me draw that. So the matrix is this nice blue area within. That's where the Krebs cycle occurs. And then the electron transport chain happens in the inner membrane space. So it kind of just happens like within these folds. Not quite outside the folds, not inside the folds in the matrix, but it happens within these folds. All right, we are finally on Unit 4, almost done. So Unit 4, we talked about the structure of DNA. DNA stands for deoxyribonucleic acid. Okay, deoxyribonucleic acid. That is the genetic code or the user manual, the instruction book for how a cell makes the right proteins to allow tissues and organs to work properly. Without DNA, the cells wouldn't know how to function. The body wouldn't know how to function. We would probably cease to exist. Okay, so the structure of the DNA is important. It's a double helix, so it's a double-stranded helix. Okay, and the DNA itself is made of nucleotides. Okay, and nucleotides are made of 3 specific parts. There's a sugar, there's a phosphate, and then there's a nitrogenous base. The nitrogenous base being adenine, thymine, cytosine, and guanine, or guanine, however you want to pronounce them. Okay, so the nitrogenous base is what makes each nucleotide different from each other. Either it's an A, or a T, or a C, or a G. Depending on what it is, changes the shape of the molecule, changes how many bonds, hydrogen bonds, that can be made. Okay, and within the double helix, we know it's made of nucleotides. Okay, the sugar and phosphate of the nucleotide is the backbone. And then the nitrogenous bases are the latter steps. And the strands are anti-parallel. On the next slide, I'll show you an image that'll make these make sense. Some things to remember is the molecule shapes for these nucleotides there's two different names. There's purines, which has 2 rings, and pyrimidines, which have 1 ring. And you'll never see 2 purines bonding together. You'll never see 2 pyrimidines bonding together. It's just not how the molecules are shaped. So if you look at these images, you'll see that our nitrogenous bases are adenine, thymine, guanine, and cytosine. Okay? If you look at this image here, you'll see that A's always pair with T's and C's always pair with G's. Apples are on trees, cars are in garages. Okay? But because of that, you will see that adenine, or our A's, is a purine. It's got 2 rings. There's a ring here, there's a ring here. Versus thymine only has 1 ring. And when these 2 bond together, they create 2 hydrogen bonds. Okay, any kind of a bond with like dashes versus a solid line means it's a hydrogen bond. Okay, and if you look at the nitrogenous bases below, you will see that this cyanine only has 1 ring. It's a pyrimidine. Versus this guanine has 2 rings, which is a purine. Okay, and when C's pair with G's, just because of how the molecules are shaped, it ends up making 3 hydrogen bonds. So A's and T's have 2 bonds. 2 bonds. Okay, and then C's and G's end up making 3 bonds, 3 hydrogen bonds. And then you'll also see that these strands are anti-parallel. One goes from 5 to 3, and the other one goes from 3 to 5, or 5 to 3, but in the opposite direction. Okay, and the directions are important when it comes to how the strands are built as far as leading strands and lagging strands, and I'll go into that very soon. So the scientists who discovered DNA, there's Watson and Crick. They're kind of jerks who stole a lot of information from other scientists, but were there and were the first to publish and describe the structure of DNA. They took information from Rosalind Franklin. She was the first to discover the double helix shape through X-ray crystallography. There's Chargaff. He created Chargaff's rules that says A's pairs with T's and C's pair with G's. And then we've got Linus Pauling, who discovered each strand was anti-parallel, which is why we end up having a leading strand, which is continuous, and a lagging strand, which has Okazaki fragments. During DNA replication, the main purpose of it is to make more cells. So in order for you as an organism to grow from a baby to a full-grown adult, you need to make more cells. The purpose of cell replication is also to develop or to replace dead and dying cells. If you give yourself a paper cut, well, your body is going to remove those dead cells and create more cells to fill in that space to create, skin that looks like it never had a paper cut before. And every cell has a full copy of your DNA, and so DNA is replicated before cell division, so each cell gets a copy so it knows how to function. So before the cell divides, it ends up having 2 pairs of DNA. So steps for DNA replication is step 1, the helix untwists and the separate DNA strands separate. Step 2, new complementary base pairs, A, T's, C's, and G's, pair up with the original parent strand. Then we have the sugar and phosphates attached to create a new daughter strand's backbone. Then step 4, that backbone is actually bonded to the bases. And then step 5, the new daughter strand is proofread to ensure there's no errors, and the strands twist back into a helix again. So basically this whole process is untwisting and separation of the original parent strands. New base pairs start attaching themselves, complementary base pairs start attaching themselves to the parent strands. The sugars and phosphates make a backbone. That backbone bonds to the bases, and then eventually it's proofread. The strands completely separate from the 2 original ones, and they twist back into a helix again. So then we have leading versus lagging strand. I keep bringing this up. The leading strand is created continuously. So when the original parent strand, this being the original parent strand, unzips, okay, there's typically a protein here called helicase. The helicase will unzip the helix, okay, and it moves in one direction. It goes to the left, okay? And just because there's a, they're anti-parallel, the DNA polymerase will attach to this end and start making a new strand in a continuous direction, chasing that helicase. Okay, so because the DNA needs to be written in a specific direction, this one is able to just continuously chase the helicase as it unzips the parent strand. However, the lagging strand needs to be built in the opposite, the anti-parallel direction. Okay, so you can imagine that as the helicase unzips, it's only going to unzip like this much at a time. So then our DNA polymerase is going to attach here and build as far as it can. Okay, as the helicase unzips more, let's say it unzips this much more, the DNA polymerase, once it's done with its first segment, is going to detach and hop here and build as far as it can until it runs into its last segment that it just made. As the helicase unzips more, okay, let's say it unzips this segment, the DNA polymerase is going to attach here and build again until it butts in to something that it's already made. So because of that, the lagging strand is discontinuous, and these little segments that are being made are called Okazaki fragments. All right, so now that we know how DNA is structured and generally how we make new DNA, let's talk about transcription and translation. So some people have a hard time remembering which one comes first. Translation has the word late in it, so it comes second, okay? So transcription is the first in the process. And what transcription is it's creating or writing a script of mRNA that's going to leave the nucleus. I've mentioned that DNA is double-stranded, so it's a little bit fatter, it's a little bit wider, it can't really fit through the nucleus pores. However, mRNA can squeeze out of the nucleus because it's one strand, which makes it a little bit skinnier. And RNA, the differences between RNA and DNA is RNA is a single strand. Its A's actually pair with uracil, and its C's pair with G's. So there is no thymine in this. It's the T's are replaced with the U. So mRNA is made similarly to DNA replication. The RNA base pairs with their complementary DNA bases to make the mRNA strand. So if our DNA was AUG, TAC, GAT, we would then use the RNA codes to pair with that. So remember, there are no T's here. So A's now pair with U's. So our A would turn into a U. A would turn into a U. Our U would pair with an A. Our G would pair with a C. Our C's would still pair with, or our T's would still pair with A's. Our A's would pair with a U. C's would pair with a G. G's would pair with a C. A's would pair with a U. And T's would pair with an A. So notice in our mRNA strand, we have no T's. Okay, this strand is then able to leave the nucleus and can find a ribosome in the cytoplasm to get translated into a protein. So what is translation? Well, translation is the translating the mRNA into amino acids, which then combine to form a polypeptide chain, which will ultimately become a specific protein. And again, translation occurs in the cytoplasm on a ribosome. So when we converted that DNA into RNA, we read it in sets of 3s. And those sets of 3s are called codons. So each group of 3 mRNA codes for a specific amino acid, which will ultimately create a specific protein. So the ribosome reads through and translates the mRNA until it finds a start codon. Then tRNA, or transfer RNA, will bring amino acids to their corresponding codons and drop off the amino acid to start being connected until the ribosome reads a stop codon. So tRNA will continue to bring amino acids based on its codon, and then once it finds a stop codon, it will stop. Okay, so this is all protein synthesis, this whole process. It's the creation of proteins through transcription and translation. Okay, remember synthesis means to form, so if we're saying protein and synthesis, we're forming proteins. Okay, and again, translation is late, so it's the second step in protein synthesis. So here's a visual of it. We have our entire cell, again, the mitochondria and the Golgi apparatus and stuff have been removed, but we've got our nucleus here, okay? And the DNA is thick, it's little chunky, it's not going to fit through. So the DNA goes through transcription within the nucleus, where the DNA is separated temporarily, the RNA strand is being made, and that RNA is called mRNA, messenger RNA. This messenger RNA is able to squeeze through the nucleus, where it goes into the cytoplasm, attaches to a ribosome, and that mRNA, this big messenger strain, okay, is going to be read by the ribosome, and these things called transfer RNAs, they kind of look like T's. These transfer RNAs will bring in amino acids based on the codons being read, and those amino acids will eventually form a polypeptide chain, which will eventually fold and contort and whatever into a specific protein that will do a specific function. So cells, just like any living thing, needs to be able to grow and develop, and some cells divide quickly, but they should only divide when told to do so. So think of the cells in your mouth, or your skin cells, or your hair cells. Those reproduce quickly. If you were to have a Hot Pocket and burn the crap out of your mouth's going to be injured for a little bit, but give it just a few days and the dead cells will get absorbed and new cells will come back, so that way you have a nice, happy, healthy mouth. Same with skin cells. If you were to fall in the gravel and scuff up your knee, it's going to take a few days to, a week or two to make that injury look like it was never really there. Your hair is constantly growing. You get a haircut 6, 10, 12 weeks later, it's just as long as before you get a haircut. That's because those cells are reproducing quickly. However, there are cells that reproduce very slowly, if at all past their first formation. So you've got nerve cells, brain cells, some eye cells. Once they're damaged, oftentimes they don't reproduce or replace themselves, which is why if you get in an accident and it damages nerves in your back, you might be paralyzed permanently for the rest of your life. Brain damage is permanent, so that's why concussions are dangerous, or certain kinds of drugs can be dangerous because you're doing permanent damage to your brain that your brain may not be able to recover from. Some injuries to the eyes lead to blindness, and once you've damaged some certain cells in the eyes, they won't replace, so you're always going to be blind. So cells can divide depending on what type of cell they are, but they should only do so when told to. Uncontrolled cell growth causes diseases and cancer. So if you have a skin cell that has uncontrolled growth, you might notice, a raised mole or a change in color or something like that. And that might be a warning to you that you have something irregular going on and you should get checked out by a doctor. Uncontrolled cell growth can cause, tumors, diseases, cancers, things that I've talked about. So cell division occurs in two major phases. So in order for cells to divide, it occurs in two major phases. The first being interphase, the second one being mitosis. So interphase is the cell preparing before division. Okay, and the preparation happens in 3 stages. There's G1, which is the primary growth phase. cells grow to a mature size, so to like adulthood in cell world, and make some centrioles, an additional organelle that isn't really in the cell until it's about to divide. Then we hit S phase, and DNA is replicated here. This is the synthesis phase. DNA is synthesized or created. So each chromosome is replicated, and now the cell has 2 complete sets of DNA. And then G2, which basically double checks everything, grows a little bit more, and then just prepares for mitosis or division. Then we have mitosis, finally. Mitosis is ultimately the cell dividing. Okay, so there's several steps to mitosis. A lot of people abbreviate these to PMAT. The first one being prophase, which is basically where the chromosomes become visible in the nucleus. The nucleus actually starts breaking down here. And once the nucleus is broken down and the chromosomes are all visible, they go into metaphase, which is the chromosomes line up in the middle or at the equator of the cell. I like to think of this as M is for metaphase, M is for middle. So that's when you know the chromosomes line up in the middle. Then we hit anaphase, where the chromosomes are split into sister chromatids. So those X's are now being turned into little ones, okay? And they're starting to be pulled to each side or each pole of the cell. Then we have telophase. This is where the cell membrane begins to start to show that pinch. The nucleus of each individual cell starts to form around both groups of chromosomes. And then we have cytokinesis, which is where each new cell completely separates from the old one. So the pinch is finally completed and you have 2 identical cells. The nucleus is fully formed and chromatids unfurl into chromatin. So here's a visual of the entire cell division process. We first have interphase, which is our G1, S, and G2. Again, G1 is our major growth phase. The cell's building all the organelles it needs, it's filling out with cytoplasm, it's getting as big as it needs to be able to divide. S is where synthesis, DNA is synthesized, so you end up having 2 sets of DNA. And then G2 is last minute growth, creates these little centrioles, which are then used during mitosis. Okay, so then mitosis, which is the rest of this, everything on this side, and that is when prophase is where you have your chromosomes form. Those are these X's. Your chromosomes are your X's. Okay, so all of the DNA from interphase condenses into chromosomes. Once your chromosomes are fully condensed, then we hit metaphase, where the chromosomes start lining up in the middle. Again, M stands for metaphase, M stands for middle. So you see them start lining up along the equator of the cell. These centrioles send out little spindles which attach to the middle of all the chromosomes. And then during anaphase, you start seeing these spindles pulling the chromosomes apart into sister chromatids. So they're no longer X's, they're half X's. Okay, so this is having one whole copy of a DNA go towards one part of the cell, and the other whole copy of the DNA going towards the other part of the cell. And then we have telophase and cytokinesis. During telophase, you can see that the cell membrane is starting to pinch off, trying to make 2 individual cells, but it's not quite there yet. The spindles go away. We start seeing the nucleus reforming on each side of the cells. And then eventually we hit complete cell separation, and we have 2 identical daughter cells in cytokinesis. All right, so that's all we've really needed to talk about for this whole semester. If you have any questions, send me an email. If you would like elaborations on certain things, send me an email. If you notice maybe I screwed up saying something, you could send me an email and let me know, whatever you would like. Hopefully this has been useful to you. Hopefully the Ed puzzle helped cement certain things. That way when you take the final tomorrow, you're going to do awesome. All right. Well, that's it for now. I hope you've enjoyed your semester for summer school. If not, I apologize, but You know, it's a weird summer school. I'd rather have it in person anyways. But yeah, so if you have any questions, let me know. Otherwise, have a good rest of your day. All right, see you guys.